And also, okay.
So we don't need to record. Did I do that or did Pritam do that?
I did that. Pritam asked me to do it, so
Yeah.
I did it.
So I'm looking for your other screen here, Pritam,
Mm.
to share with you. Oh, there we go. Guest.
Okay.
Yeah.
Good. Here we go. Let's start. And also thanks for Henrik and Susan joining this talk.
Oh, we're very curious.
I'm too afraid now. I'm like too nervous now to speak.
You have to switch the Pritam. Or is it just me?
No,
We
we're
see-
seeing, yeah, the presenter view, not the presentation.
Okay. You cannot see the presenter view, right?
Yes, this is the. On display, no, on top you see display settings, Pritam.
Where? Oh, here.
Yeah, precisely. And then swap the view and
Yeah,
the slideshow.
that's good, right?
There you go.
Okay. Thank you, Sven. So yeah, so I'm Pritam. I'll describe myself a little bit later. First, I introduce the topic. So working on environmental DNA with presence absence data, I'll describe about everything in details because we have a broad audience together, and we'll go from a beginning to the advanced level. So that's why I named my presentation as environmental DNA from presence absence to species interactions. This journey was super. I mean, with my like PhD journey and then in my postdoc, I have seen this developing field growing day by day. And I'm really excited to explore more in my postdoctoral researcher with George and Rosie. Thank you for hiring me here. I'd like to describe a little bit about myself. So I'm Pritam Banerjee. My main skills are like in research interests are environmental DNA and plant animal interaction, metabarcoding and population genetics. I also love to do teaching. I did teaching in college for 1 year. I had my private institution in India, or more than 100 students I dealt with. So that's one of my interests after research. And also I'm interested in wildlife biology, kind of like plant collection, doing herbarium things, field study, all of those together. So this is my hobbies are life, or I don't have any extra hobbies. Let's start with details of the things that we begin to understand the general diversity of the things. So why we have a diverse community living in our whole world. We have a microbial diversity, we have a macrobial diversity, and everything together. And where we also have a free DNA living around us that we came to know for a long time, and then we started to exploring that where those free DNA are, how they're moving around our world, and how they're interact, how they're showing us the signature of species present and other things. So they also sometimes like free DNA. There is also sometimes free RNA. They bound with each other. And I also believe that kind of like this cycle around the nature quite a few times. Then you can see that the different molecules cycle around the and species when they form, they after their death, they cycle around to nature again to form another species. So that we can describe a little bit later. So this is the overall diversity, and if we compare the biomass, we can see this around. So I'm excited to talk about environmental DNA. To give a brief introduction of environmental DNA, what is. So species where they leave some traces of things. Like human, where we leave some traces of things like pollutions. So species, every wildlife species, on everyone who is living around there, they leave some traces of DNA around themselves, and that can be tested out, that where species is living and how they are working. Thus, all the DNA we collected from environment is called environmental DNA. It's not only the things you're seeing that as a DNA is like a single DNA stand or something else. It more than a single DNA stand in the environment. So we'll go in the details of that one. So when species are living in environment, they have like their skin cells, they have their secretions, they have their here first, and when they also compete in competition or like interaction, they lose some of their cells outside. When, like, for example, they fight with each other, they lose some cell in the environment. Also there are some kind of like eRNA that is secreting during their interaction with environment. So each of those are living in the environment, but we need to know how to collect them and how to analyze them. So once we started doing different kind of environmental DNA, we started with like a brief collection from water environmental DNA. I'll give you a big shot of like a overall history how we started. But we have a different kind of DNA moving around us, from soils, from water, from like vegetations, like from plants, and also some like scat. In other samples, they have traces of DNA of many species living species that we can detect. But what we were doing before eDNA. So we are also doing traditional monitoring to identify species, and both the traditional and the eDNA monitoring have some negative and positive things to deal with that we came to know in details recently. And we know that some of the studies, they show that environmental DNA can detect more species diversity, and some of the studies they said, oh no, both are detecting equal species diversity. Some of the studies show that traditional monitoring detects more diversity than environmental DNA. And combining all, we can see that if we combine all the monitoring together, we can maximize the detection of diversity as well as the interaction. So each method, they might have some negative and positive views. So we need to accumulate all the method together. So in this presentation, we'll like to know all the methods of existing biodiversity monitoring and then detecting species interactions. So again, come to environmental DNA. What we need to do, we need to accumulate the DNA from environment. The accumulation term is the main thing of environmental DNA. How much we can accumulate from the surrounding. It's not like that traces of soil that we can start detecting. For microorganism is okay. For bacteria and fungi, we can do with a limitation of 1 gram soil or to like more than 2 gram or 5 gram soil. But when it comes to like eukaryotic organism, a big organism, we need to accumulate the DNA. That's why you started with water eDNA. It has become very popular because in water we can do filtration and accumulation of DNA together in a filter paper and start extracting, so that all the DNA will accumulate in a single place in the tube. But for terrestrial eDNA, the scenario is different. So we need to modification of the things. So when things of like this, we need to accumulate the DNA that they live in the surroundings, so that we can start detecting them with a PCR and other stuff. So being a very simple way that how the DNA is like, and how the thing is like, for a general idea, you can see that the free DNA is here during the 1 nanometer. And the particle DNA, bound DNA is here. And the virus and prokaryotes and other organisms, you can see the size here. So we generally use 0.45 micrometer filter that catch around these things from here to all the prokaryotes like this. They also can catch around like other unicellular organism when the filter is like start clogging, and they can catch all the cells and other things. But for free DNA, it's very important to understand how we can catch the free DNA and other stuff, because eDNA generally have many molecules together, not only the free DNA. If we go in details, what eDNA is actually, eDNA consists of organismal DNA, the cells. eDNA consists of trace elements like gametes, as I told you, the secretions, pollens, fars, spores, everything. eDNA also consists of naked DNA as well as ancient DNA, I'll tell relic DNA, that is degraded DNA, is present for environment for a long time, and also called also present as a cellular DNA. And we also call DNA from non-target organism, that another organism interacting with another organism, they have a surface DNA to carry with in different places. So they are also carrying DNA. Like in honey, we can detect from honey, you can detect thousands of plants, they are interacting with the honeybees. From mosquito, we can detect the different mammals they are feeding with, the bloods they are feeding with. Also, when flies are on something like some days ago, like Maclean came to me, and then Maclean was saying that, oh, I detected cow DNA from fly while doing non-destructive method. Oh, I call that's the perfect example of surface DNA. When they're in the cow dung, you can detect the cow DNA. So surface DNA really matters in presence of gut content also. When you're doing gut content, as Henry in this paper show that how you do bleach sterilization to avoid the surface DNA, to avoid contamination. So surface DNA also show you another kind of biodiversity. So each of the DNA here will present different kind of biodiversity to you, and you need to understand the states of eDNA to go more details. So again, there is an ecology of eDNA. You need to understand that. We need to understand before dealing with the eDNA, that is the eDNA is primary or secondary, which transportation they are going on, which state they are, how they are fed. They're facing, if there is a naked DNA, they will undergo enzymatic digestion very quickly, mechanical fragmentation very quickly. But if there's a cell, that will not go that much quickly. So we need to understand the fate, and we need to understand the transport. Transport is very specifically for if we call that in aquatic system, water will flow and you have a transport. But how about terrestrial system? Wind will also flow, you can have a transport. You need to imagine that all the things, rain will flow from the top, and you'll transport your DNA from like a from surrounding trees to the soil, and then it will transport to a nearest lake. So that's why that it is an amazing paper that Christy Diner show in the Nature Communication paper, that the river is a very important thing to carry the biodiversity of the surrounding. When they washes through rain, all the DNA will come to the river, and they will carry the river. So this is also a terrestrial biodiversity monitoring method when you understand their ecology, how they are coming through. But doing eDNA is not very straightforward. You have a different kind of like negative, positive view, yes, no view. When you need to take your decision strongly, you should include the positive samples, how many positive samples you have, how many negative samples you have, what is your control, and how you're making this environment perfect. And you need to answer all the questions. There are 14 questions, or so I forgot the 16 question you need to answer before you start environmental DNA. Doing those things. So those include control, those include organism behavior, those include how you detect the environment, which environment you're working on. Thousands of things we need to consider before going to environment. That's why when I do the literature survey and I try to understand the field, I make a clear workflow, and that I feel that need to understand in details. So this field is growing rapidly. As I say that not in every field we have seen an exponential growth of field. Like from 2008 to 2020, this field is growing exponentially. Thousands of papers are coming and different studies. And now on, we can say that from 2008 to 2023, we established the eDNA method for regular monitoring detection. But only the presence absent will not be enough for eDNA. We want more. We want to go more further. So this is the general history of eDNA. What we are doing. It's not only coming from 2008. It started from 1987, when people started detecting the first report of study of soil eDNA, when they started detecting environmental DNA from the soil, but they didn't name as environmental DNA, name as extraorganismal DNA or extracellular DNA, and that is present in the outside. Sometimes it's also affected in biofield development of organism. And from now on, we started, you can see that here, how we did, this is the first study of eDNA, where we detected the eDNA from the soil, and from the soil, we go up to the terrestrial environmental DNA. It's like, it took a lot of time, but mostly in this era of phase, we have detected many things together that we can go details. But in this slide, we need more time to present everything. But the major, most important finding here is like we have grown in much different, from soil to water, from presence absence to meta-barcoding, from meta-barcoding to species interaction. We have developed a lot in the last few years. That is rapid development that you need to catch up with all literature to start with eDNA. So I developed a really basic things that you need to understand before starting eDNA. And it took me for this figure, I study a lot, everything together. but it's very underrated. So okay, I was like promoting myself, not like this. So in this figure, I already assumed in like when I was starting doing my PhD, I thought that you can see the dash arrow here. I thought that eDNA is cycle around and transport around the environment from water to air again. That's my personal, but I try to explain from soil to air again with the flow of water, flow of air, and with the flow of transport, and from water to soil is already proven that is coming down from air to water. We already know that, yeah, rainwater eDNA is coming down, but before the rainwater publication that we have this kind of concept. I am trying to understand how this happened, and we can see this circle around. So basic few steps of eDNA, like from collection to preservation, capture, and the next few steps, you need to understand every step we named as a control. So we have a collection blank control, we have a preservation blank control, we have a capture blank control, and so on. So we have deal with so much methodology in details last few years, and those are the general methodology that we can see. But mostly this is for aquatic eDNA. I'll say there is some of the method here in NGS are like obsoleted. We can skip that one. So here is another control, that extraction blank control. And then after that, if you're doing DNA barcoding or meta barcoding, you should have a positive control or mock community or internal amplification control or internal positive control. Without those control, you cannot go to do a eDNA and say that I detected biodiversity. That will not be a solid evidence, because in eDNA we are blind. We need a support to do all those things. And from now on, then you can say that this control I have, I exclude the taxa from here, and then I detected 100% those taxa in eDNA. Also, there is some debates of doing species specific and community level monitoring. I will say that both the techniques have their own advantage of doing things. Species specific have their own advantage, community level monitoring have their own advantage in different stages of monitoring. You can give a particular emphasis on particular species to understand their expression in the environment with the species specific monitoring, and you can do a community level monitoring with targeting like whole earth about biodiversity. For example, our lab, we can say that we can only monitor a particular spider with the key qPCR and detect their level, and that is very accurate monitoring method for qPCR. But you need to maintain all the control, as I mentioned. Otherwise, it will be difficult to like deal with false positive and false negative. So around the world, we are doing eDNA for like a decade, and we are developing the things very rapidly. So we have rapid develop kits now to survey eDNA, so that a normal person or undergrad can also go into some places and collect the eDNA. They don't need to think about that one. They have a kit to collect the eDNA. And after that, they can take this filter paper back and they can do in the lab filtration, and all the workflow are standardized. And we are working perfectly on developing this throughout the world, especially when you go to tropic. The temperature and the environmental condition is different there. So once what you are doing in Europe and other states, you have a cold temperature constant, the DNA degradation might. I'll show you some result of tropics, what I did in there in the like say 35 to 40 degree temperature in hot water, in drainage water. So before that, we are doing rapidly for eDNA, and we are arranging so many conferences to like catch up with all the ideas we are developing. So attending conference in eDNA, doing research collaboration, publishing rapidly, because people are publishing everything very rapidly. It's very necessary to thrive in this field and to know, understand in details. So being another overall thing is like we have different type of methodology and different type of kits are used. So in 2029, and then in Tuji and Kumar, they did a systematic review and meta-analysis of different type of marker we are using in eDNA, as well as different type of methods we are using. I will suggest that this paper is really nice to go with. Most of the cases, some recent publications say that eDNA is more focused on mitochondrial sequence and only COI or cytochrome B. Will say that this is because of reference database. We need to build our strong reference database to deal with this sequence, because once we get data, you can do multiple things. I'll show you the different use of multiple and double primer, how they can go with. So this is the general workflow of what most of the study in before 2019 deal with fish. because it's easy to collection there. At the end, they have external fertilization, they have secretion and other stuffs, and filtration and other equipments. They are most common in there, but now scenario have changed. Now we are moving this eDNA to terrestrial ecosystem, but still we are following some general methods to developing. Now in our lab, we are developing leaf litter eDNA samples method, flower eDNA sample method. We are going to fruit eDNA samples method. We are washing the trunk roll or bark to take out the DNA from the tree. We are also going. We will also go to like leaf eDNA samples and throughout the plant and terrestrial values. But we have a more details description for that. So again, to be a very quick, false negative and false positive, you need to understand and you need to implement all the control. Otherwise, the experiment, you cannot go a specific conclusion with eDNA. It's very tricky. And as in my personal opinion about the fragments of eDNA, I'll always like to understand the difference of fragment rate, and then understand the variation of like how species in eDNA is differentiated from a shorter fragment to longer fragment, which one is more persistent to stay in the environment, and which one is not that much persistent. So that things also matter. What other matter things is like sampling strategy. You need to understand the organism biology, what they are doing. If some frog is dealing with somewhere else and you cannot collect the samples from, like if nowadays we understand that this water will flow, but if for terrestrial ecosystem, especially I'll say, if some species are only above trees diversity, they are dealing with here, you cannot samples like in the bark and other places. So you need to understand the species biology. If any of the species dealing with a different habitat and they are having a different ecological niche, you cannot sample in other places. You need to, you can sample in that places to see that if they are present or not, but you need to spread your sampling strategy to build up. So it's all the strategical game that we need to play with eDNA. So again, we have a traditional monitoring, visual survey. We have a breeding, like how we need to understand the vocalistic call out, like acoustic sound, and we have a larval surveys, and we have a eDNA. So all of the studies, like some of the studies, as I show you, they showed that eDNA, visual survey, and all of the survey are equally effective to detect biodiversity and maximize biodiversity. As I told you, many literature also proved that eDNA is more effective, and many literature for the traditional method is more effective. It depends on how we are dealing. When there is no reference database, traditional method will be more effective because eDNA cannot detect without reference database. But where is the reference database? Traditional coupling with the traditional method will be a more option to detect the maximum biodiversity. So here is another example of like how multiple primer will maximize your detection ability. If you target multiple primer with a multiple location, that can maximize your detection ability of species. But also you cannot skip traditional monitoring if you have if you do not have a perfect reference database for building with that. But skipping into there, I like to describe a little bit about my project. Yeah, this project I don't want to describe. I started this project in my PhD. This project somehow failed. I could not do it. And I'll describe personally to you why I cannot do this one at the beginning in 2019, but not in front of everyone. I don't want to do that one. So skipping to next project. Okay, so for species specific, I started doing a species specific monitoring. I need to understand the different stages of monitoring. In silico test of primers, in vitro test, mesocosm test, and also field test. These stages are important for species specific monitoring, and we did all of the things with apple snails in Taiwan, dealing with tropic temperature, excessive algae in the field. You can see my watercolor here. I did filtration of this water in algae, and then excessive amount of plants and other stuffs there. We started doing these things, but we haven't started doing with the only field survey. We developed this one in the lab with in silico PCR, then in the mesocosm with the aquarium and other stuffs. Then after standardizing, we extracted all 20 to 30 different snails taxa and then tested our primer with those taxa. And after every confirmation, like working 1 year with the lab, then I started to move on, because we need to start a species specific assay for development. We haven't done qPCR for this one because the snail was abundant. We wanted to develop a quick method with cPCR, because people can implement in anywhere with a low cost to detect this apples, and those are very invasive. I'll not go with the details of this thing, what I did in population genetics and other stuff. I'll rapidly, because I have a lot of things to go with the eDNA species barcoding. So I'm very good at field. I can jump anywhere and I can go anywhere in the details, and like to do field survey of like eDNA, and I like to take students to go their field and see what's happening there rapidly, how they're interacting with them. Those snails have two different species living in the same place, and they have a sympatric distribution. So we collected this water for eDNA, and every time we filtrate with the protocol I show you, that 0.5 million, we have a filtration equipment. We filtrate this one with positive control, negative control, 3 sample replica, everything. We started to understand the diversity of the snail, whether we can detect those snails from a pond. So we started with the small aquarium, and we go to the like a huge amount of water to detect like this field, like liters of water. We need to accumulate those DNA of the snails in that water. So field surveys are quite important. Sometimes we set up our lab in the field to do field extraction, and this is how we can you can see that how we are doing the field extraction, and it's very quick with this method. And then we preserve the filter paper like this, and we take it out. We use silica gel so that we don't need to store in the cold temperature or anything else. We just carry with the bag. And sometimes I already do sample by myself, only a scooter. I like to take a scooter and everything in a bag. So it's completely a versatile field lab. And sometimes I like to throw my undergrad in like drainage system like this, so that they can take samples, and it's very cruel sometimes. And I also like to pull up them from the system. So yeah, it's kind of fun in working in the field. I really like to enjoy the field time. So we survey all over the Taiwan, every locations. We set up each and every county. We surveyed at least 3 locations, and we compared with the 2 different base pair of primers. One is 666 base pair. That seems quite long, but we detected every samples like this, over 90% detection. And then we have our own design primer, 171 base pair primer. So we tested in Mesocosm, and also we tested in the field for detection of those primer. So I'll need to go a little bit quick. So if anyone interested in species specific monitoring, I'll describe in very detail. So those are all the result of different samples. So this Formaceous maculata species was not reported from Taiwan. We reported in the first time that the distribution is detected from environmental DNA. Then we took the species in the tissue derived DNA. Then we screened all the snails, and we found that the species is present, but very low in number, like in the ratio of like 200 to 10, like 100 to 5 in this ratio. So they are very rare, and we detected with the eDNA. But in traditional monitoring, we detected Formaceous maculata from only southern Taiwan. But with eDNA, we detected from many other places that we haven't detected in traditional monitoring. So going rapidly with a quick about the species interaction in rice field. We have two different kind of rice field in Taiwan. One is organic, one is inorganic rice field. So they use chemical pesticides in the rice field. So I was very curious about what is the different kind of arthropod diversity in the rice field when they start planting and after planting. Is there any changes of arthropod diversity in organic field or traditional field? So we have done all of the chemical analysis of soils, from starting to heavy metal analysis, and then water property analysis with the HANA machine and other stuffs, all the ions analysis. With this one, we also collected environmental DNA from 12 different fields and in 2 different seasons to see the biodiversity. But I'll show you the MCO data, how ugly is it with the fungi. I know that, but if you exclude the fungi from here, so this MCO data is generally showing the relative abundance of different taxa over the things. And this also showing the specific taxa we targeted, like we filtered out other taxa and we are detecting the specific taxa, but that include many fungi and some of the things that are present in the negative sample we haven't excluded. I want to show you that how the rotifers and other stuffs that present in the normal water is coming into your samples if you don't exclude the negative samples reads from your sample. So this was the overall read of insect diversity. I am not going to very details of what insect we're editing, what kind of interaction, but we also detected our snails from the rice field, and also some of the rice paste, serious rice paste in tropic. We also detected their presence. So we did a turning report analysis to understand that if traditional field are more diverse than organic field, or do they have a different structure of community. We find some difference in their structure, but I need a little bit more to drive on the data and more bioinformatics to understand that what actual difference is. So this is the basic O2 diversity. So I'll quickly move on to this one. So here, when we do the NDMS Pro, we can see that those are the green one and this one are organic fields, and those are the traditional fields. They have a little bit separate combination of diversity, but I was not very good at bioinformatics, but I could not do that much. But I hope I can do a details drive on the data to understand what happened. And I also I like to bring those filter papers. I have everything preserved in a well sequence in Taiwan, so understand this species diversity, and we have also DNA to understand with multiple primers, so that if there is a difference between rice field and traditional one. So this is the latest analysis of overall diversity and detection of rice field and traditional, how much taxa you can see the multiple fungi read there. And if we go in a little bit more details, that's I was very interested. The snails you are seeing, that Pomacea canulata, this one is the invasive one. And the two snails you are seeing, this one is the native one. So we have predicted that the native biodiversity is going to decrease because of invasive one. And we have detected Pomacea all over the field of every sample. Those include 3 subsamples, and those all include 3 subsamples. We have detected only 1 or 2 samples of native snails present in the field. But yeah, we haven't detected from other places. So that's another concern of invasion in Taiwan of Pomacea. So again, going to the main theme of that was example of my study. So what eDNA can tell us? Currently, we know presence, absence, species diversity, relative abundance, but we really know that. I was thinking, when eDNA can detect hybrid species, when eDNA can detect absolute abundance, when eDNA can detect the physiological status of organism, how they're facing in the environment. Like, for example, I'll give an example of spider. If the spider having a like some stress issue in the environment, they cannot deal. How we can detect with eDNA fast, rapidly? We need to understand the physiological status of the organism, its structure, and also breeding status. How we can do, we need to think about species interaction. We need to think about in details, so that we will not only face the situation in current level. So yeah, species interaction. I was very curious about how species interaction in the environment, detection of pathogen from fruits, detection of soil microbial fauna, how species interaction. But mostly species interact with something that is beneficial for them, that is like some food source. So when I was studying, before going to leaves, uh, in the leaf they have some leaf miner and some other species. But if I was thinking, if I go to fruit, they have a great source of things that insects are attracted with, and they are coming to. But in fruit, not only insect, when fruit have a strong pericarp outside, small insect cannot go inside. If this is not ripe, and they cannot feed, I mean, they cannot like eat those stuffs. So that large mammals, or sometimes like birds, they break that pericarp and open that fruit out, and then insects are going to have have a good food source there. Bacteria, fungi, they're going to have a natural food source there, and they're attracted with that one, and they go there. So changes of community in the food is really one of my interests. So I was like sitting in my lab, and there was a papaya tree outside of my lab, and I was seeing the papaya tree, one small squirrel, that Taiwanese squirrel, they're going there and eating the papaya. I was thinking that how about extracting that DNA from there? Can I detect it something? Can I detect something? Yeah. So that's all. These things are going in my mind. Oh, environmental DNA, we can detect it, detect from soil, multiple function, pollinator, and a lot of things. But in reality, they are a little bit different. So I started writing a review again. How we can detect the species diversity with eDNA? What is the main limitation I need to understand before going to the theories? But always very important to me to go in a practical stage. So I understand what is the limitation of each and every step of the eDNA for detecting a species interaction. Then I jumped into an experiment with those theory. I collected several papaya, that is eaten fruit, all over like a space of university and beyond that county in Taiwan. And I tried with another food because one food is not enough. I tried with another food, the pineapple one. I collected from farmer all the eaten pineapple around the field. I want to detect what's actually eating them. I have all the additional control. With additional control, I included some fruits that is intact, not eaten or any feeding mark. So that will be my control for environmental deposition and other stuff. If the fungi or some insect are crying there or like standing there, that will be my control for that one. So we did the study with the high pressure spraying bomb to take out the DNA. I don't want to have deal with too much plant DNA, so I didn't extract that. But the spraying method is very good to accumulate the DNA all of the surface. So we sprayed around 500 ml volume of water to the food, and we started doing filtration and the normal process we established in our lab. And we went through, and then we we detected with MCO again because yeah, I was first time doing with like meta barcoding stuff at that time from species specific. So we choose the best primer available at that time and to deal with this thing. So with MCO, yeah, we detect a lot of arthropod that is fitting in next, and I was targeting some of the chordates like that squirrel. We can detect some birds. So we detected birds, arthropods, as well as some level of like other species like many Drosophila, the fruit flies, we have detected from that fruit sample. So you can see that different species of Drosophila, they have a different heats, and we can go up to species level of detection. We detected like with the 300 something base pair, 313 base pair, and we prepared a heat tree for this. So if you exclude the fungi read here, if you go details in the arthropod animal read, you can find that most abundant here is arthropod. And after that, you can see that most abundant the Drosophila, and that Drosophila taka species is very popular in there in Taiwan. And then the other relative of Drosophila, we also detected from the sample some of the species that they are eating, the rats and also the birds that are eating the, and we also did some human contamination there. But all the data I need to filter before coming to a good conclusion about things. And also again, we can use multiple primer. So this is the scenario of pineapple samples. And now I'm going to show you the fruit that is non-eaten fruits, the diversity and abundance. You can see that there's completely, I can go a little bit before, there's completely different abundance of non-eaten food. So when you're dealing with the non-eaten food, you can see that environmental deposition and some other stuffs, but you cannot go to a deep dive of like a biodiversity with non-eaten. So they actually, when we do a intervene group, you can see that there is some common structure of like taxa that is detected from all the samples, but although they have some specific taxa that's only dealing with the pineapple and papaya, and this is the non-eaten one. They have only 3 or 2 taxa that is detected. So most of the cases they have some specific taxa for the samples, and we have the you can see that the OTU diversity of like eaten fruit and then the non-eaten fruit samples. Those are the non-eaten fruit, the single one. Okay, I'll quickly skip. I'm running out of time. So this I'll skip again. There's a diversity is completely different for non-eaten food for the eaten food. Yeah, I'd like to skip it. Here's the result of a publication from Henry and the lab. They also say that there is a different diversity of community in roots and flowers, as well as if we can do in fruits, as well as leaves. There will be a different diversity, and we need to detect those diversity in details. So I like this study very much, and I was talking to Span that give me your favor. I want to put this one there again. Oh, so here I want to tell that in general, if we are doing what we are doing is doing it with single primer. Multiple primer detection, it's very efficient for like confirming your status and other things. What they have done for single species, they targeted two different region of COI and nuclear region, and then they detected the detection abundance was like double than actual detection rate. So this we need to also use for multiple primer metabarcoding assays to be more precise for our community level biodiversity monitoring. So kind of species interaction we detected from eDNA so far. And this is the spider interaction from like a Span's work, how they detected with the gut content. I have something very interesting I want to skip quickly. So there are different methods to dealing with the insect biodiversity in general, the bug DNA biodiversity, but there are other methods to deal. There are few new methods people are working on, like that different type of traps. Here's snails, for example, but vacuuming samples, vacuuming specimens is also collecting their surface DNA, as well as tree rolling, as well as rainwater, spider waves, tea bags, flower eDNA. I'll include fruit eDNA here. Okay, herbarium samples, as well as I was discussing with Rosie last time, vacuuming of things like from air and then other stuffs is very important to detect a lot of biodiversity and their trophic interaction. Moving to next level, we have a different sampling method for different things, but what we need to do, reference data collection, standardization, upscaling with like latest technology, and also integration with different methodology and different samples. Also with multidisciplinary approach like computer vision monitoring. I was reading some conference paper of George from machine learning to DNA barcoding, that conference paper. I forgot the conference name. Yeah, so this kind of multidisciplinary approach like machine learning, radar system, molecular methods, DNA barcoding, you can need to implement all together for a special biodiversity detection up to species interaction. So how we can do that one? We only focusing on a single tropic interaction from producer to primary consumer with the flower eDNA. But I'll say that if we can standardize this one from like my ideas, like from flower eDNA to a particular species gut content, excluding surface DNA. If you have included the surface DNA, you cannot go to a next tropic level. So excluding surface DNA, the gut content of every primary consumer sometimes, and the also secondary consumer, exclusively gut content. So we can understand a correlation between sorry, I would like to tell this one a little bit later. This one, where is that one? This one, yeah. Sorry, we can understand a correlation between that. Actually, if we study the gut content and then from producer, we can study the surface DNA, plants and other animals. Then we need to go to a gut content analysis of each and every species together without surface DNA, as well as we have some other concept. Okay, so there are some good examples of tropic interaction study. Here I would like to introduce the environmental RNA for going a more one fed later. So that's really great. And for Hawaii samples, I like the Nathalie work, how they include the arthropod abundance and different field. That's really excellent to understand the different biodiversity, like species interaction, native and non-native, but how we can go more further of that one. This also, I really like the gut content analysis of the samples from Suzanne work, that how they implement, but how we can go from more than this one. It's very interesting. Yeah, this one is very interesting for do interdisciplinary approach of this one. So recently, this paper also published about gut content of mice and rats, how they did the gut content analysis and detected lots of different plants they are feeding, how native, invasive, and mixed one, and their different gut content analysis to understand that what they're actually eating, the structure. Okay, so coming to an end really about the things is like how we can go one step more further with RNA analysis. eRNA can show you the current expression they are doing in the environment, not only their presence. So if one species is doing a competition, another species, they are secreting some RNA, they're doing some this expressing some genes. They're expressing not only housekeeping genes, some genes for attack, some genes for defense. Those also can be detected from the environment in the nearest atmosphere. So we need to implement the eRNA and eDNA together to provide the ENS concept for biodiversity monitoring and tropic interaction and identification. So eRNA is also developing as a method. So we need to understand the method is similar with eDNA, but a little bit different. We can have a special discussion on that. But what eRNA can change here is like what eRNA can change. Last one was for eDNA. This one is for eRNA. eRNA can show you the gene expression. For example, someone is in the flower. They are having eating something, or they are two flies are competing with each other. They can express their gene there, and that will come out also eventually with the cells expression that we can also detect. And also during the herbivory, what kind of, and also the gene to gene concept, as we know that the species plant have some defense gene, insect have some attacking gene, how they're expressing together is very important. I like to drive more details that we can do. And after that one, surface DNA is also very effective to understand the defense and social behavior and other stuffs that we need to implement for in future. So all the concepts are coming in the end, as to say as a thank you. Yeah, I like to describe more, but I think that running out of time, we already have 1 hour. So I like to speak more about the RNA and this discussion, but I feel that that can be a second part of this one to go dive in more details. Thank you for everyone.